We will now begin with how protein structures can be determined by NMR. Before we start looking at actual structure determination, the first one step we have to do before that is called resonance assignments. So, this is the flow chart shown here for protein structure determination in general. So, we have gone through this step in the last few classes. We looked at isotope labeling and how sample is prepared. Then we looked in the first part of this course how to collect data, what are the different 1D, 2D, 3D NMR data that we collect, how they are collected. Now, we will see that once you get those data, how do you actually assign a protein. So, this is a very important step here. Assigning means that the chemical shift of every hydrogen, carbon and nitrogen in the protein should be known, means what it belongs to, which amino acid it belongs to, what is its chemical shift and so on. So, that is very important to know, and that is not a trivial step. It takes lot of time, energy and effort to do this. Sometimes you may not be able to achieve success here at 100%, typically if it may be even 60-70%. So, achieving 100% success is very difficult, especially if the protein is very large in size, and therefore, because we have a lot of peaks which are missing, sensitivity of the signals are low and so on. So, it is very difficult step here, and once we achieve this, then we go to the next level or next step where we actually solve the structure. So, we will come to this once we have understood how we can assign a protein. So, let us see again what is the assignment procedure, what is to be done in assignment. So, first thing you have to know is the exact sequence of the protein. So, as I said, this has to be given to us, we should know this correctly. So, what is a protein primary sequence? A protein primary sequence is a linear sequence of amino acids joined to each other by the peptide bond. So, these bonds are peptide bonds. So, you can see here a schematic drawing of some protein. So, these are different amino acids. So, one thing of course, you notice in this protein is that some of the amino acids are repeating. For example, leucine is coming twice in this chain. Similarly, you can see lysine is coming twice. Okay. So, like this many amino acids are not just twice, it can come multiple times. So, therefore, our goal in a assignment, resonance assignment, is to assign the chemical shift of each peak in our spectrum to the respective amino acid type. But not only that, we need to also tell that this chemical shift is for lysine number 17, this is for lysine number 24, like that. Similarly, this is the chemical shift of leucine number 16 is so and so, chemical shift of leucine number 12 is so and so. So, we are not only just telling leucine, we are also identifying which leucine in this sequence in the order that we are chemical shift belong to. So, therefore, we should have the information of the order, means the sequence, then only we can start working on the project and then try to figure out which amino acid and which glycine, leucine etc. have what chemical shifts. So, this is therefore one confusion which many of the beginners in NMR have. They ask the question, why do you need the sequence? But sequence is needed, as we see it as we go along, you will see that we need this to assign the protein. So, this how do you obtain the sequence? Sequence typically is obtained by various biological and chemical methods, which we cannot go into in this course, but I would refer you to some biochemistry book where you can see how the sequence of proteins are analyzed. So, typically if you know the genome, the DNA of a cell, you know that if you know the open reading frame, or you know which gene corresponds to which protein, from the gene sequence you can then extract the sequence of the protein. So, this is one of the methods. The next thing which we should know for assigning a protein is that where do the chemical shift come generally. So, this idea one should have very well in the mind. Okay. So, for example, in the proton, this is we are looking at a 1D proton spectrum. So, if you look at a 1D proton spectrum, typically the aliphatic groups will come somewhere here from 0 to 2 or 3 ppm. So, these are all the beta protons of methyl of the amino acids, gamma protons of the amino acids. Similarly, methyl groups will all come somewhere between 0 to 1 ppm, and they are very easily recognizable because they are very strong. Then you have this region between around 3 to 4 ppm or 3 to 4.5, where the H alpha of the amino acids will come here. So, remember the H alpha is the backbone proton alpha of every amino acid. Every amino acid has H alpha. Now, comes next after that is a huge water signal, because remember we make our protein samples in water, H2O. So, the H2O is a huge signal compared to all other signals. So, that will come here. Then comes the aromatic region, where all the aromatic amino acids like phenylalanine, tyrosine, tryptophan, histidine, etc., will come there. Now, there is the amides also start coming in this. So, amines of course, we do not worry about in protein, but the amides will start also overlapping, but slightly shifted. They come between, amides come between 7 to 10 ppm. So, they are slightly away from aromatic, although here it looks overlap, but they can be separated. But sometimes amides and aromatic will also be clashing. So, this is the typical range of chemical shifts which are observed in proteins for as far as protons are concerned. Now, let us look at carbon. Where do the carbon chemical shifts come? And carbon chemical shift comes in this range. So, typically between 10 to 45 ppm, you will get all the aliphatic C beta, C gamma, C delta chemical shift. The methyls etc., will come somewhere close to 10 to 20 ppm, 10 to 25, and the remaining all come in 25 to 45. Okay. The C alpha starts around 45 for glycines and goes up to 65. This is something which we saw in the experiment called HNCA and HNCOCA, where they come. So, this is the range typically for C alpha of all amino acids. Then some of the C betas, although C beta comes in this zone mainly, but two amino acids serine and threonine, their C beta comes between 65 to 75. And why is that? Because the C beta is attached to a OH group. So, because of that there is a de-shielding effect and the C beta comes in this range. Then between around 75-80 to 75 to 120, there is a blank region. There are no peaks in this region in proteins, and then you start getting the aromatic carbons, okay, sp2. So, aromatic carbons are coming in, they come in this region. Then again there is a gap, and then the carbonyls, that is amide carbonyls, they will come in this region. Okay. So, this is typically the range for the protein chemical shifts as far as carbon is concerned. Now, let us see for nitrogen. So, these are basically the 3 nuclei which we are going to look at in the chemical shift assignment. For nitrogen, typically it comes between 100 to 135 ppm. Okay. So, the glycines, typically the glycines, they come somewhere in the upfield regions, they come between 100 and 110 ppm, but other amino acids are all together. So, there is no amino acid specific information in nitrogen. But if you will see later that in carbon, as I said already here, for example, serine and threonine only come here for C beta. So, if I see a C beta chemical shift in this zone, then immediately I can close my eyes and say that that belongs to either serine or threonine. But how do I know it is C beta first of all and it is not C alpha? There are some ways to distinguish in some experiments. Similarly, C beta here of alanine comes in a very specific zone between 15 to 20 ppm, and again that any C beta which comes there can be immediately assigned to alanine. So, C beta chemical shifts do carry information of the amino acid type, but N15 unfortunately does not carry. So, based on chemical shift of N15 except glycines, it is very difficult to tell which amino acid that is. Now, remember also asparagine and glutamine side chains also contain amide CO and H2 groups. So, the amide hydrogens of asparagine and glutamine side chain will come in this zone between 100 and 115 ppm. So, they come at the right hand side top of HSQC. So, when we count the number of peaks of HSQC, we normally have to take into account that there are side chains also possible. So, we have to identify them and see them. So, these are the typical ranges of chemical shift for proteins as far as proton, carbon and nitrogen is concerned. So, one should have an idea of where do which peaks comes where, so that helps to assign the protein. Now, let us say we will take an example of very simple system to understand what does it mean by assignment, what do we actually need in assignment. So, let us say we have a tetrapeptide something like this, alanine glycine alanine glycine. So, this is kind of a repeat, the dipeptide is repeating. So, it is a very strange sequence, but can come in any protein also. So, let us say we have this tripeptide and now we want to assign the chemical shift. So, now you see here there are 2 alanines and 2 glycine, which means I should not simply tell that this is a chemical shift of glycine, I should also be able to identify which glycine is which chemical shift, not just saying the chemical shift is glycine some peak, I should be able to say that is a chemical shift of glycine number 2, chemical shift of glycine number 4, this is alanine number 3. So, we can actually number this peptide 1, 2, 3 and 4. Okay. So, I can write it in a single letter code like this, it is A1, G2, A3, G4, G is for glycine, A is for alanine. So, now we have to assign, let us say, this tetrapeptide. How do we do that? So, let us see first the spectrum, 1D spectrum. So, let us say that these 2 dipeptide have this following chemical shift. Suppose I make this dipeptide separately in a peptide synthesis way, I may get a chemical shift spectrum like this. So, this this is quadret belongs to the alpha of alanine and this doublet corresponds to the methyl group of alanine, okay, and this singlet is from the glycine H alpha. This is just a schematic, this is not a real spectrum, this is just a schematic drawing what you would expect to see if you had a alanine glycine dipeptide. Similarly, this alanine glycine may be, when you attach to this, okay, not separately, when you attach it may have a slightly different chemical shift. Okay. So, I am not taking the same peptide now, I am just showing you that this chemical shift of this alanine glycine may not be same as this alanine glycine, because the chemical environment is now different, there are tetrapeptide. So, therefore, it may have different values. So, when you combine this all 4 together, you may get finally a spectrum which corresponds to the full tetrapeptide, where you see here there are 2 glycines, there are 2 quadrets here and there are 2 doublets here. So, the question now is which doublet corresponds to which alanine. Okay. So, that information is not here, because when I record a spectrum of a mixture, not like this was done just to show you, but in reality when I make a tetrapeptide and I record a 1D spectrum, I will simply get something like this. So, I will not be able to tell which alanine doublet here, which of course, I know there are alanines here because I know the sequence and I can see that there is alanine in my spectrum, but which alanine peak is which alanine number 1 or 3, that information is not there here. You cannot do anything to extract it from here. Similarly, glycine, there are 2 glycines, I just do not know which is G2 or which is G4. Again that information is not present in this 1D spectrum. So, that is why this whole game of business of assignment, resonance assignment becomes important, because just looking at a 1D you do not get that information at all, which is which, even from 2D we will not get. So, we can do 3D. So, let us see at least with a 2D can we assign this tetrapeptide, how do we do that. So, let us say that we have a toxic spectrum. So, what is needed in a resonance assignment of protein is that we need dipeptide connection. So, this is something, let me go back to the previous slide. So, here you see we had the amino acid, we knew there is a glycine and alanine here. So, individual amino acid information is present in this spectrum. I know there are 2 glycine, there is an 2 alanine, so on, but I do not have a connection or correlation between the neighboring amino acid, between the neighbors, and that is what is actually needed in a assignment procedure. So, any protein assignment procedure which you will come across, remember it always will try to find a dipeptide connection. That is the heart, that is the important point in any assignment procedure. So, I would recommend you to pay careful attention to this point, that we need a dipeptide connectivity, then only I can go from one to the next and walk along the sequence. Otherwise, these are all independent amino acids I will not be able to get simply without, if there is no connectivity information given to me between neighboring amino acids. So, neighboring means one I, suppose this is I, then this is I minus 1, or if this is I, this is I plus 1. So, I need some information of connection between these two, so on. So, which experiment will give me that information? TOCSY will not give me that. So, we will see in TOCSY what do we get. In a TOCSY we simply get information of a connectivity within one amino acid. So, this is an alanine. So, this is what I showed in the 1D, there are alanine here, there are 2 alanines here. So, the one which is connected to this, its corresponding partner, it is connected with a cross peak. So, this is one spin system. This is something we have seen in the 2D part. Similarly, the other alanine also will connect within the alanine. Okay. So, this is an alanine with alanine, its own connection, and glycine, of course, there are 2 glycines, there is no connection because there are only single protons. So, you see here this TOCSY is not helping me much, it is only giving me a connection within a given amino acid, which is fine, which I need to know at least which alanine, which peak here is connected to which here. So, that at least that information I am able to get it here, which I earlier had not got from 1D. So, that is fine. So, I am able to get some information from TOCSY, and there is a transpose peak will come here, which is now shown here. So, this is exactly equal and opposite, and the same connection information is shown here, getting here. So, what do we do then to get the interpeptide connection which I mentioned? For that we need this NOESY. So, a 2D NOESY will now help us to do this assignment. So, let us see how it helps. First we need, of course, TOCSY to find out which peaks are the self peaks. Self means the same spin system. So, obviously, that is these peaks are corresponding to this here, this corresponding to that here. So, the color looks same. Remember, NOESY is not a colored spectrum like this. In reality, in practice it will come all in the same color, but I have just shown in different color to help you identify the peaks. So, you can see here now these 2 peaks are coming from TOCSY. So, all the remaining peaks are from NOESY. Of course, this diagonal is same as TOCSY diagonal. So, now let us pay a little more careful attention to these red color cross peaks. So, that is something I will show you in the next slide. So, now look here. So, basically you have 2 glycines here. Okay. So, we know there are 2 glycines. How did we know that? Because in TOCSY in a 1D, remember I told you there are 2 glycines, which from the simple chemical shift we can say, but still I do not know which glycine is this and which glycine peak is this. So, G2 and G4, that assignment is not done up to now. So, how can we achieve that is what we will see now. So, now let us focus on this glycine here. If you look at this glycine, what do you see? You see that if you go vertically or down either way, so let us go vertically, it is showing me connection or correlation to both the alanines, these 2 alanines, right, the beta proton. So, it is showing me to both the alanines. Similarly, when I go to the alpha side, it is showing me to both the alpha protons. This is alpha proton, these 2 of alanine, these 2 are beta protons. So, that means this glycine is connected to both the alanines. That where is this connection coming from? It in NOESY, how does the correlation come? It comes because of proximity. Proximity means less than 5 to 6 angstroms. So, that means this glycine, which is this particular glycine here, is showing me proximity or distance, close distance to 2 alanines, which means it could be only this glycine, because only this glycine you see is close to both the alanines, whereas this glycine is close to this alanine, but it is far away from here. So, that means now I can assign that this peak, this glycine peak is definitely G2, because it is showing me correlation to both the alanine. So, you see the neighboring information immediately helped me to say that this is glycine number 2. Otherwise, which glycine is which I could not have deduced just based on TOCSY. So, similarly, now look at this alanine peak here. This alanine peak here is showing me to both the glycines. You see this alanine. So, that means this alanine is similar to this glycine, is showing me connectivity or proximity to both glycines. that means it has to be this alanine only, because it is the one which is sandwiched between two glycines. So, it is equally close here, equally close here. So, the H beta of this alanine is having a nosy cross peak with this H alpha of this glycine, and this H beta of this alanine is having a cross peak to the H alpha of this glycine also, because they are equally close. So, you see this is how now we can clearly say that this peak which alanine, here again we had two alanines, I mean there are doublets for each alanine. So, we do not know which alanine doublet is which. Now, I can say this left side doublet is clearly the alanine number 3. So, now what is remaining? One glycine remains, another alanine remains. So, obviously one glycine which remains is this, and this alanine which remains is this. Okay. So, basically that is how we assign a protein. This was a very simple example, but you can also look at this part here. Here also if you see, this alanine is showing to two glycines. So, it is this alanine, and this glycine is showing to two alanines, which is this glycine. So, the same information, either you go from this beta proton side to alpha proton, or you can go from alpha proton to alpha proton. So, there is no issue, you can use any one of these chemical shifts to do that. So, this is a simple example to tell you that once we have a neighboring connectivity information, we should be able to connect two amino acids with each other, and with help of that we should be able to find out which glycine is which or which alanine is which in this case, or which amino acid is which chemical shift from a spectrum. So, in a 3D let us see how do we do that. So, before that, so basically if you go back to this picture and whatever we did here, we can actually see what are the two steps we did first. So, if you go back one more step, so here let me go back to this, here we first identified that there are two glycines, and we first identified that there are two alanines based on the 1D spectrum. You can use that 2D also. The first thing we did was, is summarized here. The first thing is we first identified the chemical shift, we did not assign it, we have just identified what are the amino acids present. So, I expected two glycines and two alanines because I knew the sequence. Again remember, it comes back to this point that the primary sequence should be known to us. Without knowing the primary sequence we cannot proceed. So, given that amino acid sequence was known, I knew how many glycines to expect, I knew how many alanines to expect. So, that is the first step I did, I assigned the spin system, means I identified. So, I would also write it here as identification. It is not just assignment, we are not assigned at this stage, we have identified. So, it is an identification of spin systems in a spectrum. So, which spectrum? That was a TOXI. So, once a TOXI spectrum you identify a spin system, then you can go to NOESY and then actually put the assigned or identified resonances in order. So, I can again write here identified resonances, it is not assigned yet, identified resonances in order. Order means in the correct order in the sequence. So, that second step which I showed you using NOESY is called sequential assignment or sequence specific resonance assignment, because now specifically we are telling glycine number 2 is this peak and glycine number 4 is that peak and so on. Here we did not do that, here we just simply say there are two glycines, there are two alanines, there are two other amino acid or n amino acids, but which amino acid is which chemical shift was only achieved at the second step. So, this is as far as the 2D part is concerned, means using NOESY and TOXI, and this is something we have extensively also gone through in the last course using specific peptides. We will not use specific peptide examples, we just gave a simple example. Now, let us move on to proteins, means in a larger protein sequence what do you do? So, number one is you need to label the protein. This is why because we need 2D is not sufficient like we did for TOXI and NOESY for peptides, we need 3D NMR. So, when it comes to 3D NMR, I have been telling this that we need to label the protein, we cannot do 3D NMR without labeling. So, now let us see in 3D NMR how do we analyze. So, So, in 3D NMR again this is something we saw earlier also, we have to now analyze with 3 dimensions and we use a strip plot. So, for example, you can see in a 3D NMR the base plane is first identified, this is typically HSQC, okay, nitrogen proton HSQC, and from that HSQC every peak in the third axis we get chemical shift in the third dimension. So, we draw a strip, of course, this is not physically done, this is done by a computer. A computer will plot a strip from the 3 dimension based on what you choose here, and the strip plot will be displayed to you on the screen. And based on many strip plots in the spectrum, you can then start assigning the protein. So, let us see how we can do that with 3D NMR. So, I will give an example from this very simple two experiments, HNCA and HNCOCA. So, if you recollect, basically what we had in HNCA and HNCOCA is that in HNCA we got for every pair of nitrogen and proton chemical shift, this is basically NH of a peptide backbone. So, that means for every amino acid we got 2 peaks in the third axis, and that axis consists of carbons alpha, C alpha I minus 1 and I. So, these are the 2 peaks which we saw in the 3D HNCA. So, this is a strip version of the 3D, where in the strip version for a given pair of N15 and proton chemical shift, that means for a given amino acid, remember these two combination represent one amino acid, we got 2 chemical shifts, whereas in HNCOCA we got only one chemical shift for a given N15 proton pair, and that corresponded to the I minus 1. So, this is shown here in diagram. So, for a given amino acid I, we got correlation to C alpha I and C alpha I minus 1 in the HNCA, but in HNCOCA we got only to I minus 1. So, why do we need these 2 experiments? Because actually with most important is this experiment, but in this experiment, this spectrum, I will not be able to identify which is I and I minus 1. Therefore, to help me do that, I need another experiment, and that gives me the information of which is I and which is I minus 1. So, I need that distinction or that discrimination between the 2 peaks to proceed further. So, for proceeding further, we need to connect now the neighboring amino acids. So, we saw I and I minus 1, now we have to go to I minus 2 or I plus 1 like we did for alanine and glycine. So, that connectivity information is something we will proceed in the next class, where we will see how we can actually join the different amino acids chemical shifts along this side or this side, and then continue this assignment so that we can identify the correct sequence and identify the chemical shifts. So, we will continue in the next class with 3D NMR based assignments of proteins.